Hello and welcome to Genetically Podified, the podcast that scratches that weird GM itch on the back of your leg. I am James and I am joined as always by my glorious co-host with Roberta.
Hi everyone, hello, glorious. I don't know if I'm glorious this morning.
Oh, you certainly are.
It is a glorious morning, let's put it that way.
Not in London, mate, it's pretty dreary here.
Oh.
But you're making my morning extremely glorious.
Oh, thank you, James. You too. It's good. It's good to have you back. We haven't, for our listeners, we haven't been recording for a while. We had a break, so we're back. We're back.
Back, finally. It's been about, a month of none of this fun stuff. Moving. What's it like? Terrible.
Awful. I hate moving so much. Anyways.
So, okay, let's get going.
Before we begin, the views expressed here are our own, nobody else's.
Okay. This week we are going to be looking at the different GM technologies. So in our first episode, we discussed how GM basically is just a different type of technology that can be used for good and evil. And then we looked at some actual science and the way genes work and what they're made of and what they do.
Yes, exactly.
Yeah, so today we're going to be looking at the GM technology that has shaped our crops up to today, essentially. Roberta, do you want to talk about, just very briefly, the four of them and what we're going to cover?
Yeah, absolutely. So today we're going to cover everything that falls under the legal definition of GMOs, basically, with a little twist at the end. But basically what we're going to talk about today is recombinant DNA and transgenic organisms made by Agrobacterium tumefaciens. Is that, how do you pronounce that in English? Because I always struggle with...
You got it right.
Because in Italian it's like Agrobacterium tumefaciens. Which is probably more correct because it comes from Latin, but
yeah, that's a very good point. I'm not sure
yeah, very good point.
I think the only slight difference is the C at the end of tumefaciens. We have it as an SH.
Ah, right. Okay,
tumefaciens.
Okay. Well, I'm probably going to forget that very soon anyway. So let's just move on.
We're catering both to the English and the Italian.
Oh, yeah. Well, I'm not sure about that because not many Italians speak.
It's in English.
anyways. Let's say Italians abroad. Let's put it that way, maybe.
Yeah.
So we're going to talk about transgenic organisms made with the help, let's put it that way, of Agrobacterium tumefaciens. And then we're going to talk about the biolistic approach. And we've kind of mentioned these in our very first episode, when we talked about different technologies throughout time. And then we're also going to talk about RNA interference, which is a special one, in my opinion. And then finally, the most recent one, which is gene editing technology, most specifically CRISPR-Cas9 and what it means to everything that has been done so far and why it's been so revolutionary. And then we're going to compare CRISPR-Cas9 to another technology, which doesn't fall under the legal definition of GMOs. We'll talk about it later. I'm going to leave that.
a bit of a cliffhanger.
We'll see. Yeah, a little bit. So, do you want to start, Jay?
Yeah. So the first one we're going to talk about is Agrobacterium tumefaciens. So this is kind of where the beginning of crop genetic modification began. This is the technology that began it, in plants at least. So
definitely.
The things you need to know about Agrobacterium tumefaciens, I think just for short, I'm just going to call it Agrobacterium, which is what we just call it in the lab.
Yeah.
So essentially it's a soil bacteria that occurs naturally. And if you've ever walked past a tree and seen that it kind of has like a big sort of bloated growth around it, almost like a tumour. That's caused by a subset of bacteria that do that sort of thing, that cause these big sort of tumorous growths in plants, and Agrobacterium is one of them.
It's quite a common bacteria, isn't it? It's just normally present in the soil a little bit everywhere. Is it the reason why they do these things to plants? Is it for nutrients and for like reproductive reasons? that they actually enter the plant.
Essentially.
So it's like, I think, and tell me if I'm wrong, it's like the tumorous, structures that you see on the plant are basically the reaction to the plant of the invasion of the bacteria. Is that correct?
Sort of. I mean, it is the plant doing it. But it's this, like, what causes that growth is what makes agrobacterium so useful. So any living thing, agrobacterium, its purpose, if you like, is to live, thrive, and reproduce as much as possible. So it does that by infecting a host plant. Like when you get sick with a cold, it wants to reproduce inside you and then you get sick because of it. But what the virus, in the case of the cold, is thinking is, I found something I could grow in really, really well. So the interesting thing about agrobacterium, it's got its own DNA, like all living things, and it has the ability to put some of its DNA into the plant's DNA. And then the plant will start using that bacterial DNA to do things that are good for the bacteria, but not good for the plant.
Right, okay.
So it's using the plant's own DNA and physiology against it, essentially.
So it's basically using the building blocks of life that we've spoken about in the past to make stuff for himself. It's like a parasite. It's like a...
It is, yeah.
Yeah.
It's living in and feeding from the plant.
Off of the plant. Okay.
The tree, in the case of agrobacterium, being forced to produce loads of nutrients that are beneficial to the agrobacterium, and then the agrobacterium can just continue to thrive inside there. So scientists discovered this and discovered how it worked, and discovered that agrobacterium had this way of putting its own genes into a plant. So this is like natural genetic modification. This is, this agrobacterium was way ahead of scientists. It knew how to put genes into plants.
Yeah, so it's basically a way, it has its own way of transferring its own genetic material into the plant. Exactly. Okay.
So I think that the best way to sort of visualize this is if you make a circle with your finger and your thumb.
Okay. I like your metaphors, James. I'm every time I get really excited when we do this. Okay.
Cool. Well, we'll see if this one works. So, this circle between your finger and thumb represents the bacterial DNA. So bacterial DNA comes in plasmids, which basically just means it's circular. So you'll have seen like human chromosomes are like little pairs of lines. Bacterial DNA is all in circles. So the agrobacterium has a specific plasmid which it uses to transfer its genes into the plant. So we'll go through which parts of the finger and thumb do what. The thumbnail represents the part of the bacteria's genetic material in the plasmid which allows genes to be transferred into the plant. So the thumbnail genes put bacteria genes into the plant genome.
Right.
The thumb knuckle area is a section that is responsible for turning on that gene. So making sure that once the gene is in it actually works. In the plant, it actually gets turned on and gets utilized.
Okay.
And then the whole finger section is the stuff that makes the plants sick. It's the stuff that's really good for the bacteria, but really bad for the plant.
Right. Okay.
So we don't want that section. So what scientists realized they could do is make something called recombinant DNA, which is essentially just changing DNA, essentially. In this case, it's taking out the nasty bits and putting in bits we actually want.
Okay.
So what scientists did is very easy to visualize. Basically, now change it from a circle between your first finger and your thumb to your middle finger and your thumb.
Right, okay.
So now you've got all the core bits of the bacterial plasmid which are useful. So you've got the nail, so it gets into the genome. You've got the knuckle area so that the genes get transcribed. But now, instead of this forefinger being the nasty stuff that the bacteria puts in there to make a plant sick, scientists have put in the middle finger, which is a whole new set of DNA with genes that we actually want in there. So this middle finger could represent resistance to a certain disease. It could represent a gene that makes plants grow faster or makes the food more nutritious. It could be whatever you want, essentially.
Right.
And then now, once you've got this plasmid and it's in bacterium, you've removed all the nasty stuff that makes the plant sick. You infect the plant because all the stuff that makes the bacteria effective at infecting the plant is still there.
Right.
And then it can still put in this new gene because all that core stuff in the plasmid that puts the gene into the chromosomes, plant chromosomes, and also turns those genes on and gets them going.
Mm-hmm.
Those are still there. And then you've got a plant which is making a new gene that scientists have put in there. And that's basically how it works.
Yeah. Should I try and summarize this? So I think that the reason why this was so helpful is because the agrobacterium was an organism that was already working with plants, basically. The takeaway that I want to get from all of this is that the thing that scientists used is the method that the agrobacterium uses to transfer DNA. It has nothing to do with the disease of the plant itself, because basically bacterial DNA is very versatile. It's very simple to edit and to modify because it's quite small. Bacteria don't have as much genetic information as we do. They don't have chromosomes. We have lots and lots and lots of DNA to work with. Bacteria have these very small circular molecules of DNA. And seeing a bacteria that does that basically, I guess, allowed scientists to say, oh, he does that. Can we do that as well? And basically take the mechanism, synthesize our own bits and pieces, put them together like Legos, and then say, okay, here you go, bacteria. Now, please transfer that into the plant that I want to transfer it to. And it works. And it's kind of amazing, I think.
Yeah, it is. Another way I've heard it being described is if you think about the plasmid, when it's got those nasty genes that make the plant sick and then you remove those genes and put something that makes the plant better, it's the equivalent of you've got a car and the car itself functions and it works well, but inside you've got a drunk driver which is going to cause damage to society or something.
Right. Okay.
And now what scientists have done is remove the damaging part, so removing the drunk driver and then putting in something useful like an ambulance driver or something like that.
Okay.
So the car is still the same. The mechanism by which the system works hasn't changed, but you've changed a core bit from something that's damaging and dangerous to a plant to something that's useful to a plant or to humans.
Yeah, that's a very good example. And I actually wanted to mention something about this because the legal definition of GMO says that a transgenic organism, a genetically modified organism, is one whose modification, of which the modification doesn't normally naturally occur in nature. But actually, Agrobacterium tumefaciens has been modifying other, let's say, superior organisms for a while. They found out not very recently, but like a few years ago, that a naturally occurring genetically modified organism that was modified by Agrobacterium was the sweet potato that we all eat every day. In my case, almost every day. There are some genes from Agrobacterium that were transferred into the sweet potato, and the sweet potato as we know it is like that because of this naturally occurring thing. So it makes us think also of what is natural, in a way.
Yeah, exactly. Cool. So that's Agrobacterium. And like I said, I've been using this in the lab. It's still a good technique for making it work.
You work with this, James.
I do, yeah. To be honest, it's a bit of a pain, but it does work. It works.
Okay. Yeah, right, I guess the thing that we skipped is that there's a lot of steps to this. Process of like modifying, creating the plasmids and blah 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 blah. But yeah, we're not probably going to go into that detail, are we? Shall we move on to the next?
Yeah, let's get some biolistics. Get some gene guns up in here.
Pew pew. Okay. There was more Star Wars than I intended it to be. Anyways, so yeah, biolistics. Basically, take everything you've learned about plasmids and agrobacterium, take it out of the agrobacterium and use a different way of transferring these plasmids into the plant, and you get the biolistic approach. So it's called biolistic because the way that the plasmids are transferred into the plant cells is by what we like to call a gene gun.
Yeah.
So now, a gene gun looks nothing like a gun, I can tell you that, because if you Google it, you Google biolistic approach and gene gun, it's just a huge machine that has nothing to do with guns. But basically the way it works is that in the lab, what you can do is you can create multiple copies of this tiny circular DNA. And then what you can do is take teeny tiny particles of, I think gold is very common, or maybe copper. I think I've heard copper as well at some point.
Okay, I don't know about that, but definitely
not
gold
sure about,
is the common one.
Yeah, so they are basically nanoparticles. They are super, super tiny particles. And what you can do is put them in a solution that contains these plasmids. And this particle will absorb some of these plasmids. Absorb meaning they basically stick to the particles. And then what you can do is take those particles and literally shoot them into plant cells. Now, you were telling me earlier, James, if you work with agrobacterium, you're going to infect pieces of leaf and like plant tissues and stuff like that.
Well, you usually spray the whole plant. So you'll grow a plant to the point where it's starting to make seeds.
Right.
And then you typically, either spray the whole plant or dip the plant in liquid which has the agrobacterium living in it. And then the agrobacterium infects the developing seeds. So then you harvest those seeds and some of those will have the gene you want in it.
Yeah. Well, with biolistics, you were telling me that it's more like a tissue culture, like a cell culture sort of thing, where you basically have, you said stem cells. Is that what you said?
Yeah. I mean, for simplicity, they are essentially stem cells, but for plants.
Right.
We don't call them stem cells. We call them like undifferentiated cells because they have lots of differences. But for all intents and purposes, what they do is stem cell like.
Yeah. So what you do is shoot these cells with the gene gun. It shoots them really, really fast. Let's put it that way. The particles that will make tiny holes into the outer membrane of the cell without actually killing the cell. And so you can then recover the cells that have incorporated the particle with the DNA in it. The cell will eventually get rid of the particle and keep the DNA.
So the next thing we're going to look at is RNA interference, or RNAi for short. This was a pretty cool thing because it won a Nobel Prize, essentially. The really revolutionary thing about RNAi was that with things like agrobacterium and biolistics, what you're doing is putting new genes into a plant or an organism, but there wasn't really an effective, direct and precise way of turning off the genes that are already in a plant. There are methods of doing it, but they're not very precise at all, and they can take a while. So agrobacterium, for example, would put in gene X. Gene X doesn't live in the plant, but we want it in the plant. But there wasn't a really good way of saying, okay, plant has gene Y in it. We don't like gene Y. It does something that's not useful, so we're going to turn it off.
Yeah.
And RNAi was a really brilliant way of being able to turn those genes off. So the kind of process we're looking at is, like we discussed in the second episode, talking about the importance of DNA and proteins, there is a process by which DNA codes for proteins. And we didn't mention this briefly because I didn't think it's particularly important to understand it at the time. And I won't go into too much detail now either, but between DNA and proteins, there is like an intermediary form, which is called RNA. And there's different types of RNA, but we'll just keep it simple and call it RNA.
Yeah.
And essentially, RNA interference, like it says in the name, interferes with RNA. So it prevents the RNA from making a protein. So the interesting thing about this is that one of the problems with the sort of old school ways of breaking a gene, which is essentially to mutate them, what that ends up doing is that gene is still turned on, it's just messed up now. And like we were discussing in the second episode, with proteins, it's all about structure. So if you've mutated the gene that you don't like, it doesn't do that job anymore because you've changed its structure, so it does something else. But there's no really good way of seeing what it's now doing. So it's not doing its original job, but it could be doing anything else that the scientists don't know about.
Yeah, I mean, it's basically like having a protein that is accumulating or is just sitting around and you have no idea if it's doing something good or bad.
And that's why you need food tests and safety tests, like you do with all food. But it's just that idea is that there's a big element of uncertainty behind it, in that you've turned off that gene, it's not doing that thing anymore, but you don't actually know what it is doing now. And there are other problems in terms of proteins are the most common things that cause allergies. So you could accidentally create an allergen and all sorts of stuff that just, there's negative consequences potentially. I think it's very rare that you do get one of these nasty consequences and they'd be picked up quickly, but
it is very rare. But to rule it out, it takes a lot of time and a lot of money for scientists to actually make sure that it's safe. Because of course, if it's not safe, it's not going to be commercially available ever. So RNA was kind of a cheap way out in a sense.
So, like with Agrobacterium and Biolistics, it's got the same sort of plasmid idea. You put in a plasmid into this plant either by Agrobacterium or Biolistics. But again, going back to this sort of circle feature, what we've done with the forefinger or the middle finger is put in a new section of DNA that will integrate into the chromosome and become part of the plant's genome. And that makes something which interferes with the RNA. So that RNA doesn't end up making a protein. So this way, you haven't affected the gene you want to target. You haven't affected that gene's nucleotides or its actual DNA. The gene has stayed the same. You've not made a weird protein, but you've stopped that protein from being made. Exactly. And it's just like a neat way of controlling specific genes. And again, another really important thing about this is how precise it can be. Because what you put in this little forefinger to control that particular gene is extremely specific to that gene's RNA. So this thing you put in there, this new genetic tool, won't interfere with the RNA of other genes. It will only interfere with the RNA of the gene you want it to. So it's really targeted.
Yeah, it's like 100% compatible with that strand that we were talking about earlier. This technique basically will produce an RNA molecule that will interfere, that's why it's called RNA interference, with the gene that you want not to work, so that it will not go ahead and make the protein.
Yeah, so that's RNAi, a very, very useful tool.
Yeah.
We're about to go into the final technology, but it's worth pointing out that this final technology is very recent. And Agrobacterium started in the 70s, I think?
I think the first recombinant DNA in general, studied, was very early on in the 70s. And then it was applied to crops later on. But one thing that it might be useful to point out is that recombinant DNA is being used a lot also to produce a lot of pharmaceutical compounds. So, for example, that we were talking about earlier was a bacterium, which is called Escherichia coli. Although in English, I just find it really funny because it's E. coli. I just find it so funny.
It's a lot easier to say, definitely.
So E. coli is a really common bacterium, and it's one of the first organisms to be modified with this technology. And one of the first things that scientists try to do, because it's a really important thing, and it used to be very expensive to produce, was trying to make bacteria produce insulin. And since then, that's what they did. They don't produce it with E. coli anymore. They produce it with yeast, but it's the same thing. So that gives you an example of what you can do with agrobacterium technology. A lot of the commercially available GM crops on the market are either made with agrobacterium technology or with Biolistic technology. Like you said, you're still working with Agrobacterium, James, so it's still really, really common.
Yeah, it is, yeah.
RNAi came on a little bit later, but there are commercially available fruits that were modified with RNAi. So I'll give you an example of RNAi technology applied to food that actually is commercially available, in this case, in Canada and the United States. And that's the Arctic apple. So the company that produced the Arctic apple is a small Canadian company that spent a lot of money on this. They invested basically, the family money all went into this dream of making life easier for producers. You know, have you ever had those sliced apples in a bag that you can just grab at the supermarket and have as a snack when you go around? So Arctic apple makes life easier for producers, and they also make them last longer on the shelf. And also at home, when you bring them home. It's a more of an aesthetic sort of thing. The apple is not more nutritious or anything like that. It's more of an aesthetic thing. And it means also that the apple will last longer on the shelf. So it's quite an interesting take on it. But the reason why I said this was a really good example to then talk about the next technology, which is gene editing, is that as soon as the company found out about gene editing, they're now making all the other ones using gene editing because it's easier. So they've now replaced RNAi technology with gene editing. So gene editing. And this was very swift.
Smooth.
Smooth. So basically what gene editing is is a very, very new technology. And by very, very new, I mean 2013, I think. But since then, it blew up. CRISPR now has its own journal, and there's been a lot of publications and different ways of using gene editing, not only in crop science, but also in medicine and stuff like that. But that's a different story. So I'm going to talk specifically about CRISPR-Cas9 because gene editing encompasses a lot of other technologies that have now been replaced mostly by CRISPR-Cas9, just because CRISPR-Cas9 is cheap, it's easy, it's quick, and it is the best thing we've ever had to do genetic modification. So CRISPR-Cas9 is a way of cutting and pasting genes inside of a plant genome or an organism's genome very quickly and very precisely. So when we were earlier talking about agrobacterium and biolistics, when you insert those genes into a plant, what happens is that you don't exactly know where that gene is going to be inserted. With gene editing, you know exactly where you want to put it. Now, the molecular process is very complex, and it has to do with the memory of bacterial cells from virus attacks. And there's a lot of very, very good, I'm not going to go into the details of that because there's a lot of very, very good material and videos about the process of how CRISPR-Cas9 works. And actually there is even a song, and there's a lot of official reliable explanations of CRISPR. But the way that CRISPR works, it allows you not only to insert new genes, but also to edit what is already there. So let's say, again, let's talk about the apple, for example. I can just very, very precisely change something in the genome and switch it off really quickly. It's super, super simple. Another thing that you can do can decide that you want, multiple copies of the same gene because you want that plant or that fruit or whatever it is to produce more of the same thing. You can just double it. And you could do that as well with agrobacterium, but this time you can decide where you want to put it. And it's basically cut-paste technology, and it's called editing rather than just modifying because it is extremely, extremely precise. The reason why it is so appealing to scientists as a technology is because, again, it's super easy to do, super quick, and it's cheap. So, yeah, there's a lot of controversy around it because, of course, what's the limit? Especially in medicine, there's a lot of ethical issues because, of course, this thing can modify anything. It can be used as a therapy, as a gene therapy. It can be used for so many different things. So there's a lot of ethical concerns, or where, like, the whole fear of designing my own baby and things like that. It is a big deal. It is very controversial. But that, I think, will be up to government and regulators to say, and also for us to make, as public, to make our voices heard in terms of what is acceptable and what is not acceptable. But and maybe if you want to add to that, please do, James.
Yeah, I've just got two very quick points.
Yeah,
so the
ahead,
first one,
please.
Just following on from what you said, like about the sort of the craziness of this new technology and its implications for society. The next 10 years are going to be really interesting in terms of the policy and legislature that goes on around this technology, whether it's called gene editing or if it gets lumped in with genetic modification is yet to be really decided. But in the 10 years after that, so we saw with the example of the Arctic apple, this was a much more expensive technology that they had to use, and it's slower and et cetera, but this was someone who used their inheritance to make it. CRISPR-Cas9 system and related systems are way cheaper, way faster, and they're getting cheaper and faster by the day.
Yeah, there is a kit that you can buy on Amazon for like £100.
Exactly, yeah. So the 10 years from today are going to be interesting for just what scientists are allowed to do. But 10 years after that, the real interesting policy and legislation things are going to be, how do we ensure that the public, because now this stuff is going to be available to anyone who has a spare 50 quid to modify anything they want. How do we regulate that? So that's really interesting, and definitely something, I mean, I love genetic modification, but it's definitely something to be worried about, I think. It needs to be
absolutely
regulated correctly to make sure that we don't just have crazy things going on.
Some CRISPR modified crops are already available on the market and have been deregulated and do not fall under GMO regulation. There is, speaking of non-browning and apples that don't brown, there is a mushroom that doesn't brown that has been modified by CRISPR and is readily available on the US market. That kind of leads into the question, what is GM and what does need to be regulated and what does not? And kind of leads into our surprise genetic modification technology at the end of this podcast, which was mutagenesis. We've spoken about this before, haven't we?
We did, very briefly on the first episode.
Yeah. Do you want to take this one, James? Or do you want me to?
Sure. We'll both cover it. But so essentially, we've been looking at these technologies. We've been going forward in time. But now we're going to a leap back to around 50 years before agrobacterium was even dreamed of. And essentially, like we mentioned in the first episode, this was essentially created because people wanted a good name for radiation, because radiation was seen as quite scary after things like World War II. So it started earlier than World War II, but it's really sort of started to kick off after World War II. And essentially the idea is that nature creates mutations. That's how things evolve. Some of those mutations are good and they go on to survive and make more babies, and some of them are bad and they die and don't make more babies. And these mutations happen naturally, but there are also ways that we can force them to happen. And the two sort of famous ones that were used for crops is either chemical mutation with a particular chemical called EMS. So you put this chemical on it and mutations appear in the DNA. So again, the mutation is just a change in that nucleotide sequence, the A, T, C, G. And the other way was radiation. So radiation can induce those changes as well. And together they form this umbrella of mutagenesis. And what scientists would do is either with this chemical or with radiation, get a bunch of seeds and just go to hell on them, go put bunches of chemical with this chemical on them or radiate them. And then all these crazy mutations would happen. And then you plant those seeds and you look at the grown plants and go, oh, that one's got something useful. Let's use that one.
You've said crazy mutations. The reason why, I guess, like something that is worth pointing out is that the mutations induced by nature most of the times are not useful. And that's the same with, like, they are random mutations. And then nature with evolution did the job of basically, again, as you said, like keeping the ones that work because they work. And that's the same process that you go through when you do seed irradiation or chemical induced mutagenesis, is that those are randomly allocated mutations. You have no idea what's going to happen. So some of them will look really crazy and ugly, and some of them will die. But then the process is just selecting, again, the ones that work, the ones that we like. Yeah,
exactly. So very quickly, it can be shown visually. If you draw like a line across a piece of paper, and that's your chromosome, that's got all the genes on it, that's all the DNA. What mutagenesis does is just put a whole bunch of lines randomly across your chromosome. And these are all the different mutations that were made by the radiation. Some of those will do nothing. Some of those will do something bad. Some of those will do something good. And you know, you find one where the plant, let's say, it makes the lemons purple, and that's cool. In fact, we can do it with the grapefruits, right? Because that is something that was actually made. So grapefruits that were just normally the sort of whitish yellow color, and they did this, and it was like, oh man, that one's gone pink. That is cool. Yeah,
that is really cool. So if you dig pink and you like pink.
and you like grapefruits, What a combo.
What a combo. They're actually, I think, also sweeter. So it made it pink and sweeter and less bitter, I think, as well. Yeah.
So let's say the pink gene that's changed, that is one of those many, many genes that were changed. So we'll put a circle around that. So now you've got this plant which has pink grapefruits, but also a bunch of other random stuff. So what you have to do is get the normal plant and breed the normal plant with the mutated plant over and over and over again until you've got a normal looking plant with pink grapefruits.
Yes.
What you've essentially done is breed out some of the other mutations that you don't want. So the tricky thing about this technology is that because at the time it was basically impossible to tell if you've left any other mutations in there, that you don't know what they're doing.
Yeah, of course.
So that's the issue, and loads of crops were made that way. a huge amount of the stuff that's in our supermarkets was made with mutagenesis. And in those crops, we don't know what extra mutations are in them.
Yeah, there is no way of knowing. There is absolutely no way of knowing. Also because we've said that naturally occurring mutations happen all the time. So how do I tell the difference between a mutation that I made and a mutation that just happened because science and because
nature.
Pollination and stuff like that?
Exactly.
Yeah, and also 90% of the wheat that we eat was mutated with these technologies, because scientists didn't have any other way at the time to improve crop productivity and crop yield, and they didn't know where to turn because people were starving, and science came to the rescue in a way.
I mean, it just raises a really interesting scientific point in that we are eating foods happily at the moment. No one's complaining about this, that scientists made, and they have no idea what they've done, really. Yeah. And it's so imprecise. There's the possibility for so many extra things that scientists didn't know they were doing. And that's fine, apparently. That's not GM, that's natural, that's cool. That's okay. That's okay to be grown organically. But now that we have things like CRISPR-Cas9, and on that chromosome line where we've circled the one we want, can literally just say, that's the gene, that's the one we don't want. It's really interesting now that is being questioned as to whether it's okay to eat or not.
Yeah, it is a very interesting thing. And again, I don't want to be in the shoes of those that have to make this kind of decisions. It made us question a lot of things. It made us question things like what is natural and what is not natural. Yeah. Is it not natural just because it happened in a lab? Or, I mean, we've spoken about agrobacterium and the way that sweet potato contains agrobacterium genes. And that happened in the past. And that's exactly what happens with GMOs. Right. So let's recap. So we've spoken about 4 different technologies. One is the Agrobacterium tumefaciens technology, which I still can't pronounce correctly in English. And then we've seen what the biolistic approach is. Remember the gene guns, pew pew and stuff like that. And then we've spoken about RNAi technology, RNA interference, which is not mutually exclusive with biolistics and agrobacterium. And then we've also spoken about very briefly, and there should be so much more that we can talk about when it comes to gene editing, but we've spoken a little bit about CRISPR-Cas9 and how and why it is very revolutionary. And if you want to know anything in a more specific way about these 4 technologies and how they actually work and why people have concerns about them and what are the concerns, or if you have concerns and you want us to address them, please let us know. Please tweet at us at GenPodified on Twitter, or email us at, oh my gosh, I don't remember our email.
I think it's geneticallypodified@gmail.com.
Great, awesome. Thanks, James.
I think.
We'll put the links.
Yeah, there'll be a link.
We'll put the links and the details down below. Yeah, okay. And yeah, thanks for listening. Thanks for being with us. If you have feedback or comments, or if you think it's great, share it. If you don't think it's great, let us know why, and we'll try to make it better. So yeah, we'll see you in the next episode. Woo. Thanks for staying with us at Genetically Podified with Roberta and
James.
See ya. Cheerio.
Farewell.